My name is Mr. Ogle Eso. Today, I'll be taking you through 2023 Waec biology practical. Yeah, these are the specimens. We have specimen A to specimen N. Unfortunately, specimen I, it is missing. Specimen A, it is a matured, fresh egg of catfish. You know catfish now. Specimen B, fresh egg of a domestic fowl. Picture of uterus containing fetus. Specimen D, LS. LS means longitudinal section. Longitudinal section of ovary of Pride of Barbados. Then E, we have leaf of Pride of Barbados. F, we have guinea grass. Guinea grass. Then we have cocoyam plants. A, we have corm of cocoyam. A, we have dry humus in the beaker. K, we have moist humus in the beaker. We have ripe orange fruits. Then M, we have longitudinal section of coconut fruit. Then N, we have longitudinal section of a fresh chili pepper. Now, I want to point at something very important. On that day, they will present this specimen before you. I want you to take note of all these specimens very well, because in why there are some certain things that the examiner actually look at. Particularly now, many of you will see cocoyam that you know cocoyam. So far you just see cocoyam, you just see cocoyam. That's the cocoyam plant. Now, some of you, if you see like, let us look at coconut now. You see coconut before you now. Coconut that is sliced from up down like this, it is called longitudinal section of the coconut fruit. Take note of all those little things. Don't just say cocoyam. Don't just say orange fruit. If you see an orange fruit now, so far you see orange, so we just say specimen egg is orange. Orange will give you maybe half mark. Yeah, it starts to be ripe orange fruit. Are we together now? Yes. Now today we will be looking at some important aspects on each specimen. Yeah, problem. Now, if you look at this now in biology questions or biology practical questions, what they do is to group some items that are synonymous. Look at this now. You will see some items that are very synonymous. Let us categorize specimen A, specimen B, and specimen C. You can look at them; they look synonymous. That is, you will see this one is talking about an egg. Is that not so? A mature egg of a catfish. This one is talking about a fresh egg of a domestic fowl. You know, it's still kind of synonymous. Look at a picture of uterus containing fetus. You can see egg of ovum of human being will give rise to fetus inside the uterus. So you look at this specimen A, specimen B, specimen C. They are still a kind of synonymous. Now, let us look at likely questions that Waec may be asking pertaining to specimen A, specimen B, specimen C. Now, number one, we are going to look at fertilization. What do I say?
Fertilization.
I will say fertilization.
Fertilization.
Yes, we'll be looking at fertilization method or fertilization process between specimen A, B, and C. Are you there? You can just, in the table, you can just write specimen A, B, C. Maybe just A, B, C in 3 rows or 3 columns. Are we together now? Now, if you look at a mature fresh of a catfish, they exhibit one type of fertilization. Is it internal or external? You know, there are 2 types of fertilization. Is that right? It is internal or it is external. Maybe the sperm cell and the egg, maybe they met outside or they met inside. For a catfish, is it internal or external? They undergo or they exhibit external fertilization. In most cases, they do not undergo actual sexual intercourse. The female mature catfish may lay eggs externally in their nest. Are you getting me now? Then the male will just come and pour a sperm over them. Are you getting me now? So they undergo external fertilization. If you see them, you just say this one undergo external fertilization. You know domestic fowl, actually they undergo sexual intercourse. This specimen B undergo internal fertilization. The C, is it internal or external?
Internal.
There's no doubt about it. Human being, they also undergo internal fertilization. Are you there with me now? Are you there with me? Yes. That is that. Second one, we need to look at mode of reproduction. Mode of reproduction is very important. What is the mode of where there are two modes of reproduction. Now let me say this. There is a difference between mode of reproduction and types of reproduction. Yes, some people you say mode of reproduction before you need to be saying sexual and asexual. Mode of reproduction it is quite different from types of reproduction. We have sexual and asexual. Mode we have either oviparity or viviparity, and I want to believe you know what is meant by oviparity and viviparity. Oviparity means all they say that lay eggs. Is that also? They lay eggs like chicken. Then viviparity means all they say that give birth like human being. They give birth through their vagina. Are you with me now? So let us group this A, B, C along with the mode of reproduction. So say A will be what? Is it viviparity or oviparity? Oviparity. That may be oviparity or viviparity. Oviparity. This one will be oviparity or viviparity. Viviparity because they give birth alive. Are you there with me now? You guys are not talking. Are you there with me?
Yes.
Now let's go. The next one that we are looking at is their gestation. Gestation. We are going to cover as many options as we can. I know if we are able to cover this, I know there is no way we won't be able to do something that day. Now let us look at gestation. What is it? Gestation means the period or time that organism use before maybe incubation or giving birth. Like chicken now, they need to incubate their eggs after some days before we now have the chicken now cross out, or they use their beak to break the egg. Then the small chicks now cross out from the egg. And for human being, we have 9 months. Apparently, after 9 months, you give birth to a baby. Is that also? So the gestation is the period of pregnancy, on the period of incubation, on the period before the chicks matured and breaks out. That's what called gestation. Now, what's the gestation period of catfish? How many days do you think a catfish used before they mature or to the fingerless? Yes. Basically, we use range. There are range because it may not be accurate. Even in human being that is 9 months. There are some people they were born 7 months. Is that also? Yes. Some people were born 7 months, 8 months. But we know that 9 months is the range that that many people can conform with. Are you with me now? Yes. So gestation of catfish is between 4 to 10 or 17. So apparently we can just say 17 days. 17 days. 17 days. Are you there with me? Are you there with me?
Yes.
What about fresh egg of a domestic fowl? You have chicken at home. I'm sure you have chicken at home. Is that also? How many days do you think? Apparently chicken 17 days. Chicken 17 days. Are you there? Chicken, domestic fowl, 17 days. I think you can jot on it. 21 days, I'm sorry. It's 21 days. The first one is 17 days, this one is 21 days. Then what about human being? Human being we have what? 9 months. 9 months or 280 days. You can write it in days, or you can still write 9 months. It's still well acceptable. Are you there with me now? 280 days or 9 months. Are you there with me now?
Yes.
Now let us look at where the womb developed. Where the womb developed. Where womb developed. Where the womb developed. Where the womb developed. Where do you think the womb develops? In catfish. Where? I mean the particular place. Now you can just see this on water. I think I have to clean some of these off so we can actually have it. I hope you have written this.
No. I want to know right now.
The ball is small, so we need to manage it. I need to manage this. Where the for specimen A at where in the water. Is that also?
Yes.
Then specimen B, that is A. Then B develop at where? Within the water. Within the uterus. Uterus. Then human being. Specimen B is what? Remind me again.
Domestic fowl.
Domestic fowl.
Fish.
It is done with
In their nest, they have their nest. Then the human being is uterus. Uterus, a nest. Nest. I hope you know that even chicken, they can just quickly look at some, are you getting me now? They can just look at some particles, some small leaves, they pack it together where they can lay their eggs. Are you getting me now? And after 21 days, then the the chicks is mature, they use their beak to break up the egg, then the small chicks will cross out. After 21, 21 days is their gestation period. Let me just think that. Now let us go to specimen C, D. Okay, we are done with ABC, right? Let's look at DE. Now, specimen D says longitudinal section of ovary of fat of Barbados. I want you to listen carefully to that. They said longitudinal section of ovary, only ovary of pride of Barbados. Why did not say longitudinal section of pride of Barbados? That day, some people will get it wrong. If they ask you to draw between 8 to 10 centimeter longitudinal section of specimen D, and you are not drawing a whole plant, your mark may be removed. This is, I you know pride of Barbados. I want to believe that this is pride of Barbados. Let me just do a kind of some sketch, a small sketch. This is just a small sketch. They have their own profile. This is not a sketch. Then they have. How do you understand? These are petals. These are petals. You need to manage the board. Apparently, you know, this is the what the hunter Abby.
Yes.
This is the what? The filament. This is the what? The stigma. This was the style. Let me put the stigma there. There's this stigma. And this is the what? The petals. Is that also? Now, this is the whole flower of pride of Barbados. Are you getting me now? What the specimen says, longitudinal section of ovary. If you want to draw the ovary of pride of Barbados, assuming they hatch, why? They ask you to draw pride of Barbados. Fine. But assuming they ask you to draw only the ovary, all what you need to do is just to draw. You can just draw the style and the stigma along, just like this. There's an initial D. Then make it seem broader. Then you can put stock. You can see this is the flower, the petals there. The inside this place is either you draw it this way. You can draw it this way. Are you getting me now? To show the ovary. Or double this line, then just make sure it has a follicle. Then this side too. Are we together? You can see now. Now all what you need to do is just to show stigma. Yeah, this one will be what? This one will be. And inside will be what? Inside will be what? Oval. Remember after fertilization that I taught you the other day, the oval becomes the seed. Is that also? Why the oval becomes the fruit? Now, this particular area, there is a junction here. There's a junction we now call the follicle. Are you with me now? Are you with me now? Yeah. So don't show this is just showing the internal section of the what? Of ovary of pride of Barbados. If Waec asks you to draw that, they know. But if they request for pride of Barbados, apparently you are going to draw it all, showing filament and that. If they are asking you to draw only, and don't forget your clarity. Don't forget your clarity, your alignment, your label, and your title. In Waec standard, they are going to mark your diagram based on this form 4 things. Clarity means your pencil will not be too dark. And in ecological diagrams, there is nothing like shading. You do not shade. Instead of you to shade, you only put dots. You don't shade. They look at the clarity. No matter how beautiful you draw something, if it is too dark, I say you are doing a creative and you are not shading, it will not help you anymore. Then you look at the alignment. Alignment means what we ask you to draw. Does it look like that? If we ask you to draw kind of Barbados now, you are now drawing a hibiscus flower. The hibiscus flower, they have almost the same structure from the ovary, but the arrangement of the pistil is different. Are you with me now? So please take note, and no matter what, even though question says draw between certain number to certain number of a particular diagram, you must put title. After you've drawn whatever you are going to draw, you now say longitudinal section of the ovary of pride of Barbados. If it's this place, we need longitudinal section of pride of Barbados. Are you with me now? Then don't forget the labels too. The labels, there are rules, garden labels. You can just see other materials for that. You will see your label has to be well arranged, and this line must not touch this. You can even label with capital letter, and it is pencil or two. It's not as if you draw a diagram, you now use your Bible to be drawing line. No, you use pencil to draw line, pencil to label and everything. Alright, are you with me now? Yes. So that's on that. Let me go to specimen. Let me just take you back to specimen A and B. Because something came to my mind again. Let's just look at A and B again. Differences between them. Can we differentiate between A and B? Yes. Specimen A is what? Mature what?
Egg of a cat.
Egg of a catfish. Versus what? That's specimen A. And specimen what?
B.
This one, you know, egg of a catfish, are you?
Yes.
And this one is what? Egg of a university fowl. Are you with me?
Yes.
Now, let's just look at the difference between them. There's no time. Let's quickly jot something down. Now, if you look at it, the first thing you look at is their sizes. Is that also? Which one is smaller in sizes? This one. Smaller in sizes. Are you? Small. Small in size. Relatively large. Relatively large in size. Or let me just dictate. So, are you there with me? And the second one now, no double layer. The structure of matured, fresh, matured egg of a catfish, there is no double layer. Just to just say no double layer. Then this one, double layer. Are you with me?
Yes, sir.
Then in specimen A2, airspace absent. There is no airspace. Whereas in specimen B, there is what? There is airspace. That is how many? How many? Then 4, egg covered with flexible membrane. Their membrane is very flexible. Their egg is covered with a flexible membrane. Flexible membrane. Egg covered with flexible membrane. But for a egg of a domestic fowl, they have a cancellous shell. Cancellous shell. And it's very hard. Because you cannot cover an egg of a domestic fowl. You can see the shell. Shell is kind of hard. Is that also? And it's hard to some extent than the egg of a catfish. Are you with me now?
Yes.
Are you with me?
Yes, sir.
So that's that on that. Now, on specimen C, are you there or you are not there?
Yes.
Okay, let's go to specimen C again. Specimen C, that is what? The picture showing fetus, in the uterus. Now, something came up to my mind again. What about if they ask you, identify methods that can prevent specimen C? In under ecology, there's a place where in secondary school they still attend, we teach you family planning and also. They can ask you, mention 3 ways by which specimen C can be prevented. Are you with me now? How do you think specimen C can be prevented? There are so many methods for preventing pregnancy, like the use of condom. Are you there with me? 2, they can use a saliva cap. They can use diaphragm. Sterilization is there, and so on and so on. I mentioned saliva cap. Is that also? Diaphragm, sterilization, they use injections. Some people use condoms. Some people use spermicide. Spermicide cream is a cream that contains anti-sperm. They use it to rub their private part. No matter how they have sex, there's no way they can get pregnant because the spermicide is like an anti, it contains a chemical that can break down sperm. They will not allow sperm to be motile and mobile for fertilization process. Are you there with me?
Yes,
Now let us go to let me just touch specimen C again. What about this question? They will ask you identify parts of specimen C and functions. Yes, let me just sketch specimen C on the board. So this is just a sketch. This is not a diagram. As soon as it's a diagram, it will be, there's no time, so I just want to make a kind of a sketch. Like this. Assuming this is the womb, your womb. Then you have assuming this is the baby, yes. Yes, assuming this is a baby that is suspended inside a sack. Are you getting me now? There are things that should come to your mind when if you are to observe the picture, because they said a picture, a diagram showing a fetus. Is that in the uterus? Is that also that's specimen? Now they will be asking you. You should identify at least 5 materials. Let's say 5 or 4 materials that can be found in this picture. Number 1, I'm expecting you to know umbilical cord. Umbilical cord is there. The placenta is around this area. Placenta, umbilical cord. Those one is 2. Now we talk about 3 embryonic membrane. There are 3 embryonic membrane. Don't forget. We have the amnion. Do you still remember that?
Yes.
Chorion. And what? The halanthus. What are the halanthus? Identify 5 medium or materials that can be found in the picture of specimen C and their function. So what is the function of the placenta? Are you there with me? Placenta, it is a medium of exchange of materials. Through placenta, a baby can exchange materials. Are you getting me? Yes. My medium it serves as a medium of exchange of materials. Oxygen, food, nutrients, and all that can be exchanged from the mother's blood to the baby's blood. Even though the blood does not meet, placenta is a very delicate structure in this picture, because it is one that permits medium or material to move from the fetus to the baby's body, to the mother's body, and from the mother's body to the what, to the fetus. Are you with me now? So it is a medium of exchange. Now let's look at umbilical cord. Umbilical cord. It attaches fetus to placenta. Umbilical cord attaches a baby from the navel, from the abdomen. Are you with me now? So he attached the fetus to the mother through the placenta. Are you there with me? Now we have 3 what's it called? Embryonic membrane. There are amnion, chorion, and what? Halanthus. What do they do? Number 1, the amnion. Are you there with me? The amnion, it serves as it provides hydrostatic protection. From the word hydrostatic means a fluid medium. Hydrostatic protection means a fluid medium. If you see a pregnant mother running, even though a pregnant mother is dancing, running, or jumping, because the baby is well protected by the amnion. The amnion serves as a shock absorber. You can write that one. Amnion, it serves as a shock absorber. No matter how a pregnant one is jumping, the baby will still be preserved in an hydrostatic condition. So 1, serve as shock absorber. Number 2, provide hydrostatic protection. Or do I say to write hydrostatic on the board? Hydrostatic. Hydrostatic. Or am I too fast? Hope you guys are getting it, have you? Yes. They provide hydrostatic protection, shock absorber for the what? For the fetus. Are you with me now? Now let us look at chorion. Chorion also has a protective function for the fetus. Chorion is in the middle. It also provides a protection and shape. Provide a shape and protection for what? For the fetus. Yes, protection and shape for the fetus. Are you there with me? Are you there? Oh, you are not there?
Yeah.
Now let's go to halanthus. The halanthus help in respiration and excretion between fetus and the mother. Yes, help in respiration, the exchange of gases, removal of waste products or injectory products from the fetus to the mother are strictly directed by halanthus. Are you with me or you are not with me? You guys should talk so that we know maybe I'm too fast. I'm too fast at this. Yes, okay. Are we at the halanthus? Can I repeat halanthus? Help in respiration and excretion between fetus and mother. Okay. Yes. Are you there with me? So that's that on that. Let us stop from that. Let's go to specimen D. If you look at specimen D, remind me of specimen D again. That is OV of what? Specimen D. What's specimen E? Kind of vowels, right?
No.
Specimen E is what? Leaf or kind of vowels. D is
This longitudinal section of ovary of pride of Babylon.
Yes, that's what I said. D is part of ovary of pride of Babylon. Now, on that, certain things should come to your mind. I know that day they will provide you the flower of the pride of Babylon, and it will be called in a longitudinal section. Either you are going to draw the whole flower, or you are going to draw the ovary alone, left to right. Now, you must think about the types of ovary. You know, even the specimen says ovary. The specimen says the longitudinal section of the ovary of pride of Babylon. Definitely, the first thing that should come to your mind it is types of ovary. There are 3 types of ovary. You should remember, we have the superior ovary, that also have inferior and what? An inferior ovary. So let me ask you this question. Pride of Barbados exhibits what type of ovary? I'm listening. Pride of Barbados exhibits what type of ovary? Is it superior? Is it half inferior? Is it inferior? It is superior. Yes. They undergo superior ovary. They undergo what? Superior ovary. And what's the reason? You know why? Sometimes we said identify the type of ovary exhibited by specimen D. Are you getting? They will not say state your reason in Roman Vico 1. Are you getting me now? They will say you to state something. They will not ask you state your reason why you pick that it is a superior ovary. On the reason, are you there with me? It is because the ovary are arranged. You know, we have, can I see explain types of ovary again? Do you remember when we talk about types of ovary that the ovary is placed above the floral wall? When the ovary is placed above the floral wall, it is superior. When it lies in a cup shape, that is half inferior. That also. When it is kind of, the ovary is now below, we cannot call it what? We cannot call it inferior. Now, let me talk about placentation again. Actually, what type of placentation is exhibited by specimen D? Now, what are the types of placentation? Just remember types of placentation. Is it marginal? Is it inside? Is it parental? Is it central? I'm listening. What type of placentation can be found in specimen D? Marginal. Marginal. They exhibit marginal placentation. Now, listen. Are you there with me? The reason why it is marginal is because ovule are arranged in a single row. Are you writing or you are not writing? It's called ovule. The ovule that is inside. Inside like this. You can see it on the board. Ovule. The ovule are arranged in a single row at the margin of an ovule. Are you there with me? The ovule are arranged in a single row at the margin of the ovule. Are you there or you are not there? Okay. Now, another thing now, they can ask you, you should, okay. But they can ask you now, mode of dispersal. Mode of dispersal. You can note it down, just like a subheading. Mode of dispersal. What is the mode of dispersal in Pride of Barbados? Is it by man? Is it by human? Is it by animal? Is it by wind? Is it an explosive mechanism? Which one is the most suitable mode of dispersal for Pride of Barbados? I'm listening.
Wind.
Wind. It's not true. I'm not saying agent of pollination. Pollination and dispersal are two different things. They are dispersal by explosive mechanism. Imagine that explosive. I'm sure you know the point of the fruit, their fruit. That one that is long. So we call it pan shake. It's there now. Like it is when it is green, but when it is dry, that turns to black. Don't you know Pride of Barbados?
We know it.
You know Pride of Barbados.
Yes.
If you go to you see there. Pan shakers, or people call it pan shaker. That's the point of Pride of Barbados. Sometimes when they are dry, they will just burst open. Something inside. Yes. They undergo explosive mechanism. Are you there with me now? Now, are you there with me?
Yes, sir.
So mode of dispersal is what? Mechanism. Now, what about their pollinators? Identify 2 pollinators of specimen D. What are they? What is the most suitable pollinator of specimen D?
Wind.
The most suitable?
Insects.
Insect. Then give your reason. Why do you think it's an insect? Insect will pollinate Pride of Barbados. Number 1, because they have a colored petals. I mean, their flower is very red. Yes, their petals is very colored and red, so it will attract insects. That's this one. Then the second one, they bear nectar. Nectar means a sugary content. Nectar, they're colored. Petals. Petals is a colored part. That red part. That red part, they do attract insects. That also. Insects is likely go to where that is colored. And when they get there, they now perceive a scent and nectar. Scent. Scent and nectar. Nectar means a sugary content. Once they are there, as they are trying to perceive the scent and also suck from the nectar, they will use their body to rub along the wall of the anther, and the pollen grain will be what? Will drop on the stigma for pollination process. Do you understand now? Okay. Let us go to the next specimen is what? Eve. So what is Eve? Leaf, Abby?
Yes, leaf of
Leaf or part of vowel. So let us group specimen E and F together. F is what? F is what now?
F is
Guinea grass.
form
F
of
is guinea grass.
no.
Yes.
F is guinea grass.
Without aswining.
Are you with me?
Yes, sir.
Now let us put leaf and specimen E, Abby, and specimen F. Is that also?
Yes.
Leaf of what? Leaf of part of versus guinea grass, Abby. Then leaf of what? Wild of Barbados. Wild of Barbados. So can we let us look at the differences and similarities between leaf of Pride of Barbados and what? And the guinea grass. And the what? And the guinea grass. So let's go. I need someone to discuss that. They are, if you look at their leaf, let us start from their leaf first. They have net venation. Nets. They exhibit net venation. Venation. Why this one parallel what? Parallel venation. I will explain what is meant by venation. If you look at leaves, there are two major types of venation. We have net venation. Leaves that are like this. Are you getting me now? This is a sample of the what? Net venation. Are you getting it? Either small or big. There are some leaves, as many as are planted, some leaves are like this. Are you getting me now? Like the leaf of a maize. When we go to mango maize, like leaf of a maize, if you look at them, maize, like corn, and yes, all those grasses that are very long, they undergo parallel venation. But like mango, all those leaves, either small or big, that are like this, they call them what? A net venation. Are you with me or you are not with me? So let us look at 4. Now, if you look at this, you see that there is air. There is air present in their body. Why this one air what? Air absent. You see a guinea grass, if you touch their body, there's something like an air, like an airy structure. Yes, that's that. Then under the class of leaf, we call this one the bipinate. This is the bipinate leaf. Why this one is a simple leaf? Simple. Bipinate, all those leaves that they have small leaf opposite side. Yeah? Are you getting me now? You will see that petal now. One leaf will be here, another one will be here, one will be here, another one will be here. Are you getting me? That they are side to side, side to side, side to side. That's what we call them was bipinate leaf. Are you there with me now? So another thing now there is branches. Branches present. There are a lot of branches here. Why this one? There is no what? Branches absent. Please take note. All the differences and similarities are very important between a leaf of part of Barbados and a leaf or let's say a guinea grass. If you look at it, so in a Waec specimen, the person you look at is grouping. It is until you group them. If you group them, you will see that they have something in common. There is no way they cannot be saying it's the difference between part of Barbados and Fuetus in the mother soup. Doesn't make sense. Are you getting me now? They rather be asking questions on specimen A and specimen B. A is egg, that one is egg, that one is almost egg. They are similar. Part of Barbados stays alone. Leaf of part of Barbados guinea grass. Right? Two of them are leaf. So expect something like this that differentiates between leaf of this and leaf of this. Are you with me now? So having said that now, what about similarities? Sometimes Waec may be funny as they're asking you for similarities and differences, they'll be asking you similarities. So that one is simple. If they ask you similarity, there's no need for tabular form. They just say both possesses this. Now they are both green in color. Is that also? They contain chlorophyll, which make them green. Are you with me now? They have a ratches. Ratches means the one in the middle. So they have ratches present in both. Are you getting me? And they both have a leaf blade. Leaf and blade. Leaf blade, ratches, leaf blade. So you can just say they are green in color, both of them are green in color because of the chlorophyll. Are you there with me? They have a ratches, both of them have ratches, and both of them have a leaf blade. Are you with me now? Okay, so let's start on that. So, having said that, after the next specimen is what?
Cocoyam plant.
G and what? G and what? So let us go to G now. Specimen G. Right, specimen G now. Specimen G. Specimen G is what? Remind me again.
Cocoyam plant.
Cocoyam what?
Plant.
Then the next is what?
Form of cocoyam.
That is H, right?
Yes.
H is comb of cocoyam. Comb of cocoyam. Let's go, shall we? Now, if you look at cocoyam carefully, I'm sure you know cocoyam. They have a very broad leaf. Is that also? Now, what is meant by comb? Comb is the stem that has been propagated. You know, they have it is their root. You know, they store their food in the stem. Tell about people, you will see their underground stem. Are you getting me? That's what we call a comb of a cocoyam. Now, what do you think? Why will they ask you from this? Now they will ask you for the ecological zone. It has to identify the ecological zone. Zone is where they are found. Like in particular zone, you know, there are savanna, we have tropical rainforest, we have this, we have that. They'll be asking you should identify the ecological zone of specimen G and what? And H. What is specimen F? Maybe you should just match it together. So F is what?
Guinea grass. Guinea grass.
Okay. Let me start with which is F. Now, what about F and G? I forgot to talk about their ecological zone. Now, what's the ecological zone of G? Where do you think G can be mostly found? The guinea grass, right?
Savannah.
No, they cannot be really found in savannah. They can be found in the tropical rainforest. Yes, if you see where there is heavy rainfall, you see a lot of guinea grass everywhere. Then what about F? F is what now? F is what?
Guinea grass.
Guinea grass. What about G?
Cocoyam plant.
Cocoyam can be found in tropical rainforest. Why F can be found in savannah grassland? Take note of that. G at the tropical rainforest, F at the savannah grassland. Yeah, you said it.
No, I don't say that.
So let us look at features of G. First, features of G. What is our feature? This one. Feature. I'm sure you know, you've seen cocoyam several times. Is that also? Number one, the first thing to come to your mind when you see the plant of cocoyam is their leaf. Is that also? Their leaves are very big, very broad. Is that also? So they have broad leaf. Are you there? G, broad leaf, broad. Broad leaves. And they have a succulent leaf. Succulent. If you look at their leaf is very succulent, very soft. Yes, very soft.
Specimen G.
You know cocoyam. Cocoyam, they are very soft and they have a very broad leaf. Broad. So, are you there with me now? So on corn is the tuber part of the cocoyam. I said it the other time. The one that's been propagated. Now there could be a simple food test on it. They can ask you when a specimen cut a small portion of specimen H Are you getting me now? Why can still ask you that? You just say specimen H is equal to a portion of specimen H and had a few drops of iodine solution. Yes, because it's a starch. If I ask you to record the observation and what? An inference, just like in chemistry. So if by venture you have a cup of cocoyam, that's the tuber part of the cocoyam, you cut small portion, you now have few drops of iodine. What do you think is going to happen? Yeah, it's going to turn to a color. Which color? You just see black coloration observed or color observed. The inference will not be what? What is present? It's a starch present. Yes, you can still ask that. So whenever you see a comb like that, or whatever it is, you know it is going to be a starch present. Are you there with me? Are you there? Are you done with that? Now let's go to adaptation of specimen H into their habitat. What do they have? How have they been surviving in their environment? Are you there with me? Now, one, are you writing? They have board for propagation. Yes. Let's go. We're already in G. We're already in H. Rather.
They have what?
Board. BUD. Board for propagation. Board for propagation. Are you there?
Yes.
They have the scaly leaves for protection. All these points are very important. We need them. Scaly leaves for protection. Are you there? Are you there?
Yes.
Now they have adventitious roots. Adventitious.
Yes.
Adventitious roots. Are you there? They are modified stem. They modify their stem for food storage. Modify their stem for food. I told you the tuber part is the comb. They modify their stem for food storage. Yes, they modify the stem for food storage. That is that on that. And don't forget what is the method of propagation. Or they can ask you, what is the method of reproduction? What is the types of reproduction executed by specimen H? I'm listening. Is it sexual or asexual? What type of reproduction? Exhibit or can be exhibited by specimen H? Asexual. Asexual through what?
Two bodies.
Asexual through vegetative propagation. By what? By comb. Is their comb. They use their comb. They use their comb for vegetative propagation. I'm saying it now, please take note of all these things. Yes. Are you there now? So let's go to J and K. Yes, we are almost running on. J, specimen J. Specimen J is what?
J is dry humus in the paper.
J is dry what?
Humus in the paper.
Yes, this one is kind of tricky. Humus. In what? In the beaker. Then the next one is what? K. K is
Wet
what?
or moist humus.
Wet or moist. They are same.
Humus
In what?
in the paper.
Humus. In the beaker. Alright? When you have a dry humus in the beaker, as specimen J, then you have another wet one in the beaker. Now, the first question that will come that I will say is this. Which one is rich in soil nutrients between J and K? Eh? Is it J and K? Which of these things is rich in nutrients? It's K. Because when water is being added into the humus, it will increase the soil fertility. It will make further decomposing process. Are you getting me now? Material or a medium will decompose faster in a wet moist than in a dry humus. You can even try a natural home experiment. If you have a normal, maybe a skin or maybe a meat of a cow, drop it on a dry humus. Then another bit of a cow, you drop it on a wet humus. Then leave it for like 5 or 10 days. Observe what is happening. You will see that the level of maggot, or the level of decomposing process in that wet humus will be more than the one in dry humus. So that's what I'm about. Now, what is the function, or what is the significance of specimen K? Are you there with me? You can write it. Significance of specimen K. Are you there? Are you there? You are not there. Now, one, it improves soil fertility. Whenever there is humus, normally soil is very rich in humus. That is why we regard it as the best soil for farming. So, improves soil fertility. Are you there? Are you there?
Yes.
It lowers the soil pH. The pH value of the soil will be lowered. Lower the soil pH. Are you there? Yes. Provide medium for microbial activities. Provide medium for microbial activities. Provide medium for microbial activities. Are you there? It provides certain elements that are important to plants. Provide certain elements that are important to plants, such as nitrogen, sulfur, and phosphorus. Yes. Provide certain elements that are important to plants, such as nitrogen, sulfur, and phosphorus. Then lastly, are you there? Lastly, the composition of humus. Are you there? The composition that I explained the other time. The composition of humus releases carbon dioxide and ammonia. And ammonia to the atmosphere. The composition of humus releases carbon dioxide and ammonia to atmosphere. Ammonia to atmosphere. Are you there with me now? So the next one is what? The next one is what? Specimen what? M and what?
The composition of what?
So we are getting to the final. Specimen M and what?
L
M and N.
L.
Specimen L is what?
Ripe orange fruit.
Okay. Specimen L is ripe. And the next one is what?
Inner section of a coconut.
That is specimen. What is that?
Specimen
M. LS of what?
of a coconut fruit.
Coconut fruit. Okay. The next is what?
Longitudinal section of a fresh chili pepper.
That is N, Abi.
Yeah.
LS of what?
LS of a fresh chili pepper.
Of fresh
Chili.
chili. Chili pepper. You can see 3 of them are still in the same group. Is that also?
No.
Because they are still group. They are still fruits. Now, the first thing that should be coming to your mind is type of fruit. What type of fruit are they? What type of fruit is orange? What type of fruit is coconut? What type of fruit is chili pepper? Now let's go. Let's say L, M, and N. Under types. Let's go. Let's go. Let's go. L is what? Fruit is what? What type of fruit is orange? Orange is a berry. Don't forget that. They are berry. Abi? M is what? Coconut is a drupe. Chili pepper is what? You see a berry. Pepper and tomato. They are still under berry. They are still under what? Berry. Now, another thing that they will be asking again is this. What type of placentation can be found in M and L? M, we have parentia. Placentation now. Parentia. And is free central. Sorry. N undergo exile. Exile concentration. Now, what type of placentation can we find in orange?
Exact.
Eh?
Exact.
What type of placentation can we find in orange? Orange in the middle. So you see exile. Exile placentation. Under placentation. Are you there with me now? Now, the major difference is now is differentiate between a coconut and orange. I think we should be able to do that.
Yes.
Yes, we should be able to do that. And I will ask someone now. What about differences between specimen L and M? L and what? And M. L is what?
Ripe orange fruit.
M is what?
Coconut.
Now, let me sound this warning. When you are trying to differentiate in biology, we are looking at morphological differences. Particularly if it's a live specimen, things that you can actually see. Don't be telling us orange is sweet, coconut is bitter. No, this is not acceptable. Because that experiment, you are not asked to taste it. So you cannot know maybe the orange is sweet or coconut is sweet. Are you getting me now? So let us look at things that you can see. Oh yeah, let's go. What can we see? The first thing you look at is their seed. Seed. Let us look at their seed first. Orange may have 2 or more seeds, or some, let's say 1, 2 or more seeds. Coconut how many seeds? Only 1 seed. There's no way coconut can have 2 seeds. Coconut only have only 1 seed. Have you seen coconut that have 2 seeds before? Coconut have only 1 seed. The one inside that we eat, that's the seed. Okay? Are you there with me now? Then what are the other things you can see about coconut and orange?
Hard surface.
Epical. This one is soft epical. Yes, and what? And epical. Yes. What other thing you can see about them?
Master, juicy mesocap.
Eh?
Juicy mesocap.
Which one is juicy?
Orange.
Juice.
The
The mesocap of orange. Do you know where the mesocap is found? The mesocap is that white one. There is one orange, the one that is green. When you peel it, you see one white. That's the mesocap. Then the inner one is one that is juicy. Are you getting me now? So it is a juicy endocarp. Are you getting me now? So they have a juicy endocarp. Now, for coconut, the endocarp is not juicy, but it's what? It's not milky. What do we call it?
fibrous.
What about in apple? I'm just citing apple as an example. Apple is not juicy. If you eat it, what do you call it? It is crispy. Yes, crispy. If you eat coconut, it will do kero in your mouth. That is called crispy. Crispy endocarp. Yeah. So, what other thing you can talk about? Yes. Let us look at it carefully. What about their mesocarp? The mesocarp is fibrous. In orange, mesocarp is soft. Why the mesocarp is fibrous? The mesocarp is what? Is fibrous. Now, the last thing now under the fruit is mode of dispersal. What is the mode of dispersal between specimen L, specimen M, and specimen N? Are you there with me? How do you disperse orange? It is through animal and man. Animal and man will disperse orange. What about coconut? Coconut is man and what? Man and water. Water will disperse it. Sometimes they fall during the rain, then the stream or river can carry them along. When they hang around the surface of the land, then they can now grow. So dispersal, man and animal. This one is man and water. Then what about pepper? Pepper now, it is Animal and what? And man. Animal and man. Animal and man. Are you there with me or you are not there with me? Now, you need to go and study their diagrams. Their diagram. That's the last thing before I leave. Their diagram. You need to learn how to draw a perfect diagram. Yes. When you are drawing your orange, make sure your orange is not. Don't use a cover of a bottle. Orange cannot maintain a perfect circle. Are you getting it? Yes. So if you want to draw, you just draw. We need ecological diagram. We need a free hand. Free hand. It has to be free hand. So people don't be looking at a cover of a bottle so that you have no. There is no way your orange can have a perfect circle. Are you getting me now? And once you have it now, you just double it. You just double the line. Just double the line. Just try to double the line. When you double the line, you can just have some mark to show the inner part. Just mark it out. Then you put the placenta. Placenta is in the middle. Then the way you have your placenta in the middle, the next thing to do is to draw your endocarp, your juicy endocarp. This is just a sketch. Then from there, you put your seed in each chamber. This is just a sketch. After that, you put something like all the juicy sac. You put something like a line to show a juicy. Yes, and your orange. Then don't forget your title. Your title will now be, is it a longitudinal section or a transverse section? This will now be what? Transverse section of what? Orange fruit. And your label, don't forget, your label is so clearly. This one will now be what? You guys will talk now. Epical. Abi. Then you have the mesocarp.
Endocarp.
Endocarp.
Then the endocarp.
Placenta and seed.
You say what?
Placenta
Placenta
and seed.
and the seed. What does that mean?
Succulent or
You can put succulent of these are the major epical, mesocarp, endocarp, the placenta and the seed. Please, all this is very important. If you are not drawing orange, you may be drawing coconut. For coconut is not hard. Just have to scale the board. Coconut also cannot maintain a perfect whatever. Just you can even bend the side a bit. Then this one, you now bend it up. Then once you have it, you draw another one. Then you later close this side. Once you've done this, you just put the inside there, then you double it. This is just a sketch, this is not a diagram. This is just a sketch. Assuming it is on drawing book, to be more finer. Are you getting it? And you just put this one showing the fibrosity. That is, the inner is fibrous. If you like, you can put small to show that there's water inside. And you are done with your coconut. You only put this way, you call it a scar. This one will be your epicarp. This one will be your mesocarp. This one will be your endocarp. This one is your seed. And don't forget, what are you going to remember again? You must put this. Is it longitudinal section or transverse section? Remember, this is longitudinal. If you are cutting a fruit from up like this, down like this, longitudinal. If you are going to slice a fruit this side, This one is transverse section. There are some fruits that if you see their diagram, you know maybe they are longitudinal. This one now is transverse section because it's being sliced. Take note to it. This one now is being cut from up like this, which is what? Longitudinal section. The title has to be clearly stated. Don't forget what I told you, your alignment, your clarity, your label, and your title. So clearly with pencil, you just go write LS, longitudinal section of what? Of coconut what? Coconut. Don't just write longitudinal section of the coconut. No. Longitudinal section of the coconut. Yes, I'm sure, having gone through all these specimens, I'm sure on Tuesday, I think, you are still going to smile and you are going to remember me. My name is Ogunle Simon. Then, see you again. Thank you.